and they just live as normal flora on the skin, mucous membrane, and even to other organisms, including the soil, microbial flora. So they are all over, and most are harmless. They are not pathogen. Gram staining. The gram stain, as we said, they are gram positive. They look gram positive, and grape-like clusters. I hope in the group that's already started practicals, you already appreciated the morphology of some of bacteria, including these typical Staphylococcus morphology, gram positive and grape-like clusters. So it's typical for staph. How do we identify a staph and how do we group them? So clinically, they are grouped in two. We use coagulase test to group them. There is coagulase positive Staphylococcus, which is only one and the most important pathogen, that is Staphylococcus aureus. And there is a bunch or a group of coagulase negative Staphylococcus or Staphylococci, frequently involved in nosocomial and opportunistic infection. So that is Staph epidermidis, Saprophyticus, Hemolyticus, and the two other Staph. So generally, we have to differentiate Staph aureus from which is a primary pathogen, but also can be a normal flora depending on where the bacteria is located. And coagulase negative Staphylococcus, which are frequently or in most cases nosocomial, so they cause infection to patients at hospital, but also opportunistic infection. So they are not primary pathogen. The primary pathogen is Staph aureus, and you have to differentiate Staph aureus from all these other. And coagulase is the most important test. You remember coagulase test is the most important test to differentiate aureus from the other Staphylococcus species. Basic characteristics include the arrangement that we said, grape-like clusters, and gram positive, of course. They are non-motile, and they are facultative anaerobes. Their colony on blood agar, maybe I show you the colony. Sorry, too much. So the colony looks like golden color on blood agar. We see that, how this looks like. Yeah. So you can see how the colony of Staph aureus looks, or all Staph looks like, with beta hemolytic pattern. I hope when we say this, you immediately understand what beta hemolytics mean. So they are hemolytic on blood agar. They have beta hemolysis, produce catalase and coagulase enzyme. This is Staph aureus. So they have these two enzymes, catalase, that differentiate them from Streptococcus and Enterococci, but also coagulase enzyme, which differentiates Staph aureus from the rest of Staphylococcus. and it's found as normal flora on the skin and nasal passage, so the nasal tract. So they are always there in most people. So as variance factors, so Staph aureus have capsule. Huh? They have capsule, which is polysaccharide slim layer on top of peptidoglycan layer, as you remember the structure. This is the cytoplasmic membrane in which there is a transport protein permease, and the peptidoglycan layer, which is white because it's gram positive. And we have teichoic acid, and this teichoic acid are also part of the variance factor. We have protein A, which is part of the variance factor. And as we know, capsule is antigenic, so they induce immune response, but also they are antiphagocytic. They inhibit phagocytosis, so they allow this bacteria to evade the immune system. We have teichoic acid, which binds to fibronectins on the surface of the host cell. So the teichoic acid, which binds on fibronectins, allows this bacteria to adhere on the surface of cell host. So the adhesion. Then we have the protein A, which is here, which has great affinity to FC portion of IG. This is one part of antibodies and inhibit phagocytosis and opsonization. I don't know, I think you haven't started immunology, but opsonization is the induction or facilitation of phagocytosis by antibody binding on the pathogen, on this bacteria. The phagocytes are able to easily internalize the bacteria, and that is by facilitation of antibodies like IgG, with phagocyte having some receptor, FC receptor, and allowing easy phagocytosis, and that is opsonization. And protein A of this Staph aureus allows escaping phagocytosis by binding the receptor of the receptor where the phagocyte will bind actually, and inhibit then the opsonization. So protein A is also like capsule, a facilitator to evade the immune system. And once the bacteria can evade the immune system, then the bacteria can divide, can grow, can produce enzyme toxin that we see now. So other variance factors or the factors include enzymes and protein that contribute on invasiveness. So they allow bacteria to go deeper in the tissue, deeper in the body. Those include coagulase. We already discussed on this, which allows clot plasma, interfere with phagocytosis, facilitate spread in the tissue by evading the phagocytosis. Hemolysin, that lyses red blood cells, leukocidin, which kills leukocyte, fibrinolysin, digest fibrin, break down fat, down the fats, and this enzyme that facilitates spread in tissue by destroying hyaluronic acid, which is part of the connective tissue. So, meaning by producing this enzyme, they allow destruction of the tissue, and thus bacteria can go in deeper. Protein A that we immediately say is antiphagocytic. So all these are part of the components that allows bacteria or Staph aureus to be pathogenic. So we have chemotaxis inhibitory protein that allows inhibit migration and activation of neutrophil. So the neutrophil are not called to the site of infection, rather they are inhibited and remain. The bacteria then can proliferate. So they have beta lactamase that is associated with antibiotic resistance, or beta lactam, and Staph aureus can form biofilms on host and prosthetic surface that evade host defense and antibiotics. When it make this biofilm, so the antibiotics cannot get in this biofilm. The larger polysaccharide thick wall surrounding the bacteria, we allow this bacteria to evade the host immune system, but also resistant to antibiotics. It protects completely the population, the bacterial population. Produce toxin that contribute to tissue damage. Not only enzymes, but it also produces toxin, like one enterotoxin, which is heat stable, which cause food poisoning. So enterotoxin is heat stable and cause food poisoning. By producing this exotoxin on foods, then the people can ingest the food which is already containing this toxin, and will cause a problem. There is another toxin, toxic shock syndrome toxin, which causes shock, rash, discoloration of the skin, we see. And there is epidermolytic toxin, A and B, which generates peeling of the skin. We see how this can really make skin to be removed, peeled. So there is three category of disease that can be caused by Staph aureus. We have pyogenic, two category actually, pyogenic disease and toxin-mediated pathology. As pyogenic disease, we can have local lesion of the skin. By local lesion, you can impetigo, furuncle, boil, and all these inflammatory superficial on the skin. Or it can cause systemic infection, like septicemia, endocarditis, osteomyelitis, arthritis, or even deeper. So pneumonia, especially in post-operative patients, and abscess. So these are more pyogenic disease, which goes with pus and can be local as well as deeper in the body, like septicemia and endocarditis. So causing treat or even death of the infected person. So as toxin-mediated disease, we have food poisoning that we already say, gastroenteritis due to enterotoxin, which lead to vomit and watery, non-bloody diarrhea, especially linked to consumption of contaminated food. So when the staph aureus divide or multiply, grow on food by producing this food poisoning toxin, enterotoxin, the individual consuming that food will definitely be affected by this toxin. Enterotoxin stimulate release of cytokines, which act on enteric nervous system to activate the vomiting center in the brain. It is heat resistant and resistant to stomach acid and enzyme. So, which is a very tricky toxin. Imagine being resistant to heat, mean that even when cook the food, contaminated food, it might not affect this toxin. And they can also resist to acid in the stomach, so they will definitely cause problem. There is types, A to F, and all are immunogenic, so we can have type the antibody. And this symptom can easily be resolved within 24 hours. So vomiting, diarrhea, watery, non-bloody diarrhea can be over as quick as 24 hours. Another toxin related disease-mediated pathology is toxic shock syndrome, mediated by toxic shock syndrome toxin, which is characterized by fever, low blood pressure, skin rash, vomiting, and shock due to enterotoxin F. So sometimes followed by 1 to 2 weeks later by desquamation, skin desquamation in palm or hands, and sore of the feet, occur mostly in women using tempo in their they sometimes individual with wounds in their patient with nasal packaging also can allow bacteria to have or colonize and produce this toxin. The toxin stimulates release of interleukin 1, 2, and tumor necrosing factor, which actually are the main cause of the symptom that we see now. So this is rash in erythematous rash. So with this toxic shock syndrome, you can see the body is just having all these kind of rash all over. So a second, beside the rash, we have staphylococcal scald skin syndrome, which is also called Ritter's disease, which is characterized by fever, erythematous rash, loss of hair and nail, mediated by exfoliative or epidermolytic toxin, which act as protease, cleave protein, which attack the epidermal cells, resulting in separation of epidermal layer to the body. It is common in children, neonates, and adults with renal failure. This is the condition that we are saying. The skin is kind of peeled from the body, and is due to the exfoliative toxin or epidermolytic toxin that destroy this protein, which is the protein that allow attachment of epidermal cells. So by destroying this protein, the body or the skin is just peeled, as you can see on the image of this newborn. So as laboratory diagnosis, how do we diagnose Staph aureus? First, the specimen will depend on clinical manifestation. So in most cases, it will be pus, swab from infected area, sometimes sputum, cerebrospinal fluid, blood, especially in septicemia. Feces can be also used, especially when suspected diarrhea or vomiting from Staph aureus enterotoxin, food poisoning, and nasal swab from carrier when you want to confirm carrier. Microscopy, you will see gram-positive cocci in cluster or single in pairs, as we previously said. In culture, you will see Staph aureus producing golden yellow colonies that are beta hemolytic, as we said. So they will look more goldish. Can see how they look. And beta hemolytic, of course, you can see a clear zone surrounding each colony. And we can also use a selective media, that is mannitol salt agar, to differentiate Staph from other microbial specimen containing, like in feces, you have to really use a selective media like mannitol salt agar with high salt, which inhibit the growth of most other organisms. We say that Staphylococcus are halophile. They can go up to 10% sodium chloride concentration in the media. And also the pH indicator in the media that allow visualization of mannitol. When the mannitol is fermented, the acid is produced, and this allows the pH indicator, which is a phenol lead, to turn to change the color in yellow. The colony will be yellow because of this acid in the colony due to mannitol fermentation, and this will be for aureus. But if the midis does not ferment mannitol, in the colony we remain the color of the media red. So, or this is mannitol pink in yellow. This is mannitol positive and mannitol negative, and this is mostly Staph aureus. Some additional biochemical characteristics include coagulase. We already said catalase allows differentiation of Staph from Streptococcus and Enterococci, but coagulase allows to really differentiate Staph aureus from other Staphylococcus. Dinase also is an important test. Dinase positive also for Staph aureus. You can also do latex agglutination to detect protein A. You can also do PCR, polymerase chain reaction, to amplify specific targets and differentiate staff from other. So there is a clinical concern. We have methicillin-resistant staph aureus, which carry multiple resistance to antibiotics, and this is very important and cause problem with treatment of staph. 95% have already penicillinase, so they are resistant to penicillin and ampicillin as they degrade this. Sometimes abscess must be surgically perforated to allow antibiotic to get in. And systemic infection require intensive lengthy therapy. As we previously said, this is bad as it will also not select staph aureus, but also kill the normal flora. And too bad. As treatment, the infection acquired outside the hospital can usually be treated with penicillinase-resistant beta-lactam. So you can treat this with but with hospital acquired, in most cases, those are resistant to all beta-lactams, and you will go with vancomycin, which is a strong antibiotics. Patient and staff carrying epidemic strain, particularly methicillin-resistant staph aureus, should be isolated as part of the control. You can also have BAST or treated with antibiotic to eradicate MRSA carriage. It's as a universal precaution by healthcare provider to prevent nosocomial infection, hygiene, and cleaning. Or it's important to really eliminate this or prevent the methicillin-resistant staph aureus dissemination. So, any question with Staphylococcus or aureus? Any question?
Question lecture. Where that bacteria host in which organ does it host in the body?
Yeah, come again.
In which organ in the body does that bacteria host?
No, ntabwo nkumva. Urabaza iki, James?
Ndi kubaza ni ikihe gice cyo mu mubiri ino bacteria colonizinga cyangwa se hostingamo?
As normal flora or pathogen or disease?
As pathogen.
So who can answer the question from James? Anyone can answer the James's question? We have seen that it can cause skin or the surface disease. In that case, it is more on skin, on the nails, and so on. But it can also cause systemic disease, where it can cause septicemia. It can go all over, including meningitis, endocarditis, and all over the body, osteomyelitis in bones. So it can reach the entire body. Is that clear to you, James? So with is it clear, James?
Thank you.
We have pyogenic disease where local lesion of skin, but we also have systemic infection, means can reach all over the body, including in the heart, in the bones, in articulation, in all over. Is that clear?
Yes, thank
And
you.
of course, toxin-mediated disease is always generalized there, as you have seen. Any other question? So if no question, let's continue. Other pathogenic Staphylococcus species will not take too much time, as you have seen. All these are parts mostly of normal flora. They can cause disease, mostly nosocomial, but also opportunistic diseases. They are not as primary pathogen. So Staphylococcus epidermidis, that is number one. Also gram-positive cocci, grape-like clusters, so you cannot distinguish it from Staph aureus by morphology. They form white-raised colonies. They are non-hemolytic on blood agar. So automatically you can see the difference. Yeah? Why this is non-hemolytic? Because it have no the enzyme hemolysin that destroy the red blood cells. They are also catalase positive, but they are coagulase negative. So to differentiate this from Staph aureus, you have to do coagulase, you have to see the hemolysis on blood agar. It does not ferment mannitol, so it will have this color on mannitol salt agar. Although catalase positive, which differentiate them from streptococci and enterococci, they will be coagulase negative, but also not mannitol fermenting, and they are sensitive to novobiocin, which is the differentiation. Its difference with another common staph, Saprophyticus, which is resistant. So novobiocin, we allow differentiation of this epidermidis from Saprophyticus, which is resistant. It is part of normal flora, skin, mucous membrane, in nose, even in the ear. So in most cases, it's hospital acquired in case it causes disease and may cause endocarditis and bacteremia following the catheter and other appliance position in the body. So you appliance or devices in the body can easily be colonized by this Staphylococcus epidermidis and cause disease from there. Also cause sepsis, mostly in neonates, and peritonitis in patients with renal failure, peritoneal dialysis. Whenever they do this, so they can lead to the infection with Staph epidermidis, which is normally found on the skin. It produces glycocalyx, which adhere to the catheter. So it adhere firmly on the catheter, and it's highly antibiotic resistant. So it has a high it has this beta lactamase and resistant to methicillin and nafcillin. So it's highly resistant, and you have to use combination therapy, vancomycin plus rifampicin or other glycosides to really treat the infection. So having this, we challenge the treatment. So the rest of diagnosis, as we previously see, you can differentiate this from Staph aureus, but also from staph saprophyticus by using novobiocin disc. Any question on staph epidermidis? I don't expect question. Huh? This is part of normal flora and can only be opportunistic pathogen and mostly following the appliance positions in body. So by themselves or normal hosts, they don't have problem except in neonates and probably in patients with renal failure, peritoneal dialysis. So when the dialysis is done through the peritoneal membrane, then it can cause decolonization in problem. And treatment is hard as the bacteria epidermidis is by naturally, or it is naturally resistant to many antibiotics. You have to do combination of antibiotics to cure. So saprophyticus, that is another staph that we need to differentiate from the two previous. Most infections are hospital-acquired, opportunistic pathogen in immunosuppressed hosts, and is strong associated with presence of foreign bodies, as we have seen in the previous. So when appliance in the body, position in the body, then this will always come in. Prosthetic heart valve, intravenous catheter, urinary catheter, cerebrospinal fluid shunt. All these kind of external bodies placed inside, we allow colonization by these saprophyticus and allows causing problem over there where it is placed. It is coagulase negative as previous, and inhabit the skin surrounding the genitourinary tract. It can cause UTI, urinary tract infection, in mostly young women, sexually active, as it inhabits the surrounding area of genitourinary tract. It can be treated with quinolone like norfloxacin, and it is always resistant to novobiocin, which allow its differentiation from the previous, the MEDs. And it is also as previous resistant to antibiotics like penicillin and cephalosporins. Hemolyticus, staph hemolyticus is have hemolytic, beta hemolytic on blood agar. It is also coagulase negative staph, vancomycin resistant, and this is one of the discs that allows its differentiation. And it is the second clinically isolated coagulase negative staphylococcus. You have staph epidermidis, which is the first, Hazaho staph hemolyticus as coagulase negative. It is nosocomial pathogen, so it is mostly acquired to the hospital. And infection includes native valve, endocarditis, sepsemia, and also mostly the external bodies, as we said. It forms biofilms, especially on these devices. And this biofilm is really resistant to many antibiotics, always multi-drug resistant. So it's hard to treat, difficult to treat. And And you have to combine many antibiotics to cure, or sometimes surgery to allow cure of the patient. The species is multi-drug resistant. So Staph aureus can be distinguished from other Staphylococcus on the basis of the following criteria. I think you already have this. One, it have the golden yellow colonies on rich medium. So they have these golden yellow colonies, and is often hemolytic, often beta hemolytic. It is coagulase positive, whereas others all are coagulase negative. And this can allows quickly differentiation of this. It also possesses the NAS enzyme, whereas other rarely possess this enzyme, and antibody to protein A agglutinate Staph aureus, but not other Staphylococcus. So by having these criteria, you can quickly differentiate Staph aureus from which is the primary pathogen from these all opportunistic pathogen Staphylococcus species. As summary, you can see we have the most frequent aureus, Epidermidis saprophyticus, and you can differentiate by base of coagulase, which is always negative, including hemolyticus, dinase, mannitol. This is positive on mannitol, sucrose positive for novobucin disc, which you have resistance, and by combining all this, and so you can be able to differentiate Staph aureus, Epidermidis saprophyticus in your growth or media. Any question about Staphylococcus? Any question about Staphylococcus? Any question about Staph? If no question, we can move forward with a second genera or a second bacteria, group of bacteria, Streptococcus, which is also a gram-positive cocci. It's indifferent to the grape-like, irregular grape-like clusters. Staphylococcus appear or have these chains morphology. Image here. This is Staph and Streptococcus. So gram-positive also in chain. They appear in chain, and this is because of their biology division. So when divided, they remain close to each other, thus forming these kind of chains. So they are non-motile, like Staphylococcus. They are non-spore forming. They don't form spore, and they are always catalase negative. So you can differentiate these to Staphylococcus by this enzyme or this test. They are also facultative anaerobes. They can grow in presence or absence of air or oxygen, and they are capsulated. some species are capsulated, some others are not capsulated. They do have capsule or not. This is same as same as staph. We have some strains capsulated, some others not capsulated. So the classification is based on two criteria or two main criteria. One is hemolysis, the ability on blood agar. We can differentiate three categories. Some are alpha hemolytic, beta hemolytic, and gamma hemolytic. And there is a carbohydrate in the cell wall called C substance. And based on the antigenic variation of these C substances, you can be able to classify groups of streptococci. So based on this carbohydrate in the cell wall, the antigenicity varies. So the antibodies can agglutinate some or not. So we have A, B, C, D. They are called Lansfield's antigen group. We see this. By type of hemolysis, as we said, you have three categories. We have beta hemolytic, which we have seen that surrounded by a large yellowish hemolytic zone in which no more intact erythrocytes are present in beta hemolytic. We have alpha hemolytic, which is more greening because of oxygenated, which converts hemoglobin to methemoglobin, oxidation of hemoglobin. And you have gamma hemolysis, where there is no visible hemolysis zone. So it's only colonies, but the hemolysis is not there. Here we have the oxidation of hemoglobin, and here we have clear zone surrounding the colony, and this is beta hemolysis. So we can classify our streptococcus based on this. Some are beta hemolytic, some others alpha hemolytic, and some others are non-hemolytic or gamma hemolytic or hemolysis. So by Lansfield's classification or antigen, we have A, B, C, D, and by alpha hemolysis, we have only one species genera, Streptococcus pneumoniae, alpha or non, virulent streptococci, beta hemolysis, group A, and this is the lead pathogen, Streptococcus pyogenes. We have beta hemolysis also for B group, Agalactiae, and all these highlight or in bold are the main pathogen that we see. So starting by group A, Streptococci or Streptococcus pyogenes is gram-positive. We have seen some are capsulated, some others are non-capsulated. Catalase negative always, non-motile, non-spore-forming, and cannot grow in the presence of 6.5% of sodium chloride. So it is not halophilic, and this can differentiate it from Staph aureus or Staphylococcus. So it is more commensals for up to 5% adult, 10% children. So it colonizes respiratory tract for all these people. And transmission is mainly by droplet. It's spread by droplet. So as virulence factors, we have quite a huge number of virulence factors. One is the F protein, and the F protein mediates fibronectin binding, which allows internalization of bacteria into host cells. So the bacteria can get inside the cell using this F protein. It also have M protein that is antiphagocytic, allows adherence to the cell, and allows grouping, group A, which is beta hemolytic streptococci, and M protein, or we also have capsid for some strains, and it is, of course, antiphagocytic. Sometimes there is a complement 5 activated peptidase, which inactivate or cleave the C5, SAS, which mediate recruitment and activation of phagocytes. So it also participates in antiphagocytic mode by cleaving the activated complement. It has streptokinase, which is fibrinolysin, which activates plasminogen plasmid, dissolve fibrin in clots, allowing spread. So when the clot is formed, the streptokinase, we really destroy this clot, and by destroying clot, then the bacteria can spread in the tissue. We have hyaluronidase, which is also a spreading factor, which degrades hyaluronic acid, ground substance of connective tissue, allowing spread of the pathogen in the tissue. It has streptolysin, which lyses red blood cells, leukocytes, platelets by forming pores in the cell membrane. It has streptolysin O, which is labile, cause beta hemolysis when colony grow under the surface of the medium. That is the cause of beta hemolytic. And streptolysin S, cause beta hemolysis around colonies on VA, cell bound, not antigenic, produced in the presence of serum. It kills the phagocyte by releasing the lysosomal content after engulfment. It has exotoxin, pyrogenic exotoxin, which produce both scarlet fever, then it acts as a super antigen. It has pyrogenic exotoxin A, which is responsible for most cause of streptococcal toxic shock syndrome. We see this condition. And erythrogenic toxin, which cause scarlet fever, see this rash. and exotoxin beta B that rapidly destroyed tissue, resulting in necrotizing fasciitis. So it's like the other powerful toxin that allows the pathology that we see with this. So there is three category of disease caused by Streptococcus. One is pyogenic, so that is more linked to bacterial growth and production of all those enzymes. So the second is toxigenic, which is more related to the toxin. And the third is immune-mediated non-spirative conditions or manifestation. As pyogenic, they start mostly with pharyngitis, as we have seen that it transmits through droplets. And they are spirative, pus, complication, tonsillitis, and abscess. So otitis, sinusitis, meningitis, pneumonia. So it can be localized or more systemic. Bacteremia, sepsemia. So it can be of constant. Toxigenic, we have scarlet fever, we have streptococcal toxic shock syndrome. So by the toxin, they can induce this scarlet fever, which we see in the streptococcal toxin shock syndrome. As immune mediated, so this is more like no longer pathogen involved, but only the immune system induced by the pathogen. So the pathogen may or not be in there, but it already induces the immune system that will cause disease. One condition is acute glomerulonephritis, which is more due to immune complex mediated disease. So the immune complex, they will be the accumulated in kidney membrane and impede filtration and cause the inflammation over there, and that is acute glomerulonephritis. And we also have acute rheumatic fever, where antibody produced in infection, in initial infection, cross-react with antigen in heart muscle. So during the initial bacterial pathogen, so they produce antibodies against the bacterial M protein or other antigens, and these antigens automatically cross-react with antigen in heart muscle. So then they will be like autoimmune. They will cause problem as they will destroy the heart muscle by just considering the reaction of antigen antibody. So entry is by respiratory always as exit, and they can spread to all over, as we said. They can cause rheumatic fever, we have seen, and glomerulonephritis, and this is non-spirative, no pus. So the pathogen may still there or not, but the glomerulonephritis, which is caused by immune complex deposit in the renal, in the kidney, and rheumatic fever, which is more caused by cross-reactivity of antigen to the heart muscles. So they can cause diverse pathology, as we have seen, pyogenic disease, where you can see meningitis, otitis, sinusitis, and all these other conditions. Some localized, but some other generalized, like on skin, impetigo, erysipelas, scarlet fever that we see, which is mostly toxin mediated. So this is, sorry, you can see how the tonsillitis or the inflammation in here, or skin, the wounds infected, or the rash, scarlet skin syndrome with red rash due to the toxin, bacterial toxin. As laboratory diagnosis, so specimen may be swab, throat swab, pulse swab, blood, depending on the infected site in the body. So you can do gram staining, direct blood gram staining, and this will be gram-positive cocci in chain, as you have seen. We have pus cells. We have culture on blood agar. You can incubate this on SAT7 only 24 hours, and you have colonies, which are beta hemolysis. And some strain that produce hemolysin O and not hemolysin S demonstrate lysis when incubated anaerobically only, as we have seen that hemolysin O is oxygen labil, can be inactivated by oxygen. So by gram stain, you will see something like this from your specimen. And on colony, you can see, as we said, they are always catalase negative. This is catalase positive, catalase negative, and this is staph. And on blood agar, they are beta hemolytic. You can see small colonies, not as staph aureus. And we have one important disc called bastracin. They are always sensitive. This is the zone of inhibition. So it means this plate was inoculated entirely by the specimen. We have the first area, we have the second area here, we have the third area, and the final, where you have the isolated colonies. And you can see that the bastracin has inhibited the growth of colonies over here. And this bastracin sensitive activity allows us to differentiate Streptococcus pyogen or group A Streptococcus from other Streptococcus that we see. So you can do latex agglutination, you can do ISO and streptolysine O test, especially in case of arithmetic fever, suggests recent infection in case exaggerated immune response to earlier respiratory infection. So the skin infection does not induce this, so they will not induce streptozidine or antibodies. So only the case of pharyngitis, the internal colonization, internal infection, we induce this anti-streptolysine O test. You can do antidinase in case of acute glomerulonephritis. So you do no longer, or possibly you don't have the the pathogen, but you can do this antibody, detecting this antibody, which is related to streptococcus by diagnosing this condition, this immune-mediated condition. So as a treatment, antimicrobial susceptibility, penicillin is the drug of choice. So far, no resistance have been seen with penicillin. Cephalosporin, tetracycline, erythromycin, chloramphenicol can be used, especially in case of internal or spread of infection. Some resistance to macrolides has emerged. You can see some. Prevention is really to treat pharyngitis, streptococcal pharyngitis, as earlier as possible to prevent all these immune-mediated disease. No vaccine available, and infection control measures can be implemented to limit the transmission between patients. Any question on Staph aureus, streptococcal pyogenes? Any question?
Question.
Yes, please.
Among the beta hemolytic, there are group A, group B, and group C. What is the meaning of those groups? What is the criteria used to classify the beta hemolytics into group A, group B, and so on?
Who can answer the question? Who can answer the question? Among the beta hemolytic? So we have beta hemolytic, which is A. We have beta hemolytic, which is B. We also have beta or non, or non, which is in C, F, and so on. What makes the difference between this and this and this? Is that your question? Is that your question?
Yes.
Anyone can answer? This is more related to antigen. Is more related to carbohydrate in the cell wall of the bacteria. So meaning that group A have a type of a carbohydrate and M protein that allows production of antibody that only agglutinates this group, cannot agglutinate this. even if this also produce hemolysin that destroy red blood cells, the group B have its different antigen from A and from C or FG. And this criteria, Lancefield criteria, is based on the cell wall components, although all are made of this M protein. The antigen that, or the antibody that is induced can agglutinate one of these and not others. And this is the base of classification by Lancefield. Hemolysis can be or not, but the agglutination means antibody antigen reaction. Here, you can group streptococci in different groups based on their components. So, antigenicity of carbohydrate, C-carb substances in the cell wall, which induce antibodies that only react with some specific category of streptococci. But hemolysis ability is a crossover. They can be in group A, but produce same hemolysis pattern as B, as C or FG here. Is that clear, Christy? So the grouping here is based on the cell wall component of this bacteria. Birumvikana, Christy?
Meaning that all different antigens cause agglutination.
Yes, meaning that in the cell wall of streptococci, there is this C substances that allows this bacteria to induce antibodies that correspond to this antigen, to the C substance antigen. And by typing these C substances, you can see the category. Nukuvuga ngo ukoresheje iyi substances, induced response is different. And the response allows typing the category of the streptococci in A, B, C, D, A, F, G category. Different from the pattern on the blood agar, the observation of how the bacteria lysis or destroy red blood cells on the media. No? Clear?
Yes.
So, no vaccine, infection control measures can be applied to reduce transmission. Group B, streptococci, or also called Streptococci agalactiae, which colonize tract, genital tract, or lower gastrointestinal tract for up to 40% of women. So it's more prevalent and can colonize neonates through mother's genital tract. They are gram-positive in chain, non-spoiled, non-motile, usually capsulated. As virulence factor, they have sialic acid that of capsules, which prevent inhibit the opsonization again, inhibit activation of complement, thus evade the immune system. To have peptides that inactivate activated complement, hemolysin that lyses red blood cells, So we see camp factor, which is done in conjunction with Staphylococcus aureus. There is others like hyaluronidase that destroy hyaluronic acid, adenosines, surface protein that binds human IgA, adenosine. So all this allows the bacteria, like hyaluronidase, allow bacteria to spread, inhibition of or evade of the immune system. And clinical condition is more cause of neonatal sepsis and meningitis, cause neonatal pneumonia, and can cause urinary tract infection in pregnant women, and endometritis, wound infection, infection in men and non-pregnant women are then older and have underlying condition. So they don't have this. So they are also opportunistic, especially for men or non-pregnant women. They can cause bacteremia, pneumonia, bone and junction infection. They can infect soft tissue, and in this case, the mortality is high, as also the infection appear to affect people with underlying conditions. So as laboratory diagnosis for this Streptococci agalactiae, specimen may be blood and so on, depending on the infected site. Gram-positive cocci in chain, and culture can be seen as small circular colonies with beta hemolysis. With beta hemolysis as identification, they are bastracin resistant. We have seen that so we have seen that pyogens or group A streptococci are bastracin sensitive, so you can differentiate them from these as these are resistant, but also production of camp factor that we see. So this is the camp test. A strict and allows to visualize interaction between Staphylococcus B lysis on erythrocyte, which is enhanced by extracellular factor produced by Streptococci agalactiae, called the CAMP factor. CAMP stands for Christie, Atkin, and Matt Peterson, who discovered this phenomenon earlier. So this blood agar plate contain 1/3 of B lysis producing Staphylococcus aureus and 2 cross-3 of the isolate. We have the Staph aureus in here, and you have the agalactiae or the specimen in test, and you can see that where the interaction or the Staph aureus interact with the agalactiae or unknown sample, we create a larger zone of hemolysis, clear zone. And this is more caused by interaction between B lysine and this factor produced by agalactiae and induce more lysis. As you can see here, you have beta hemolytic and you have beta hemolytic, but when interacted or closer to this area, there is much more wider clean zone. So as treatment or how do we manage this? There is penicillin, gentamicin, vancomycin, but it is important to always do the susceptibility testing, and know whether it's something or not. Sometimes it will require combination therapy, especially when the patient is severely ill, like sepsis, neonatal sepsis, or other advanced or problem. So you can do combination to really kill quickly the pathogen. Any question? Any question with group B, streptococci? If no question, let's have a break, a 15-minute break. 15-minute break, come back. Hello? A question?
Hello.
A question?
Yes, can you? Yes, question. Can you talk again about the CAM factor?
Who can explain the CAM factor? Anyone can explain the CAM factor? Mubo turi kumwe muri class. Hari uwamusobanurira CAM factor uburyo Anyone can explain the CAM factor? So before we go to, let's come again to the CAM factor. So this is a blood agar media in which there is a streak, a line of Staphylococcus aureus. And we know that Staphylococcus aureus produce an enzyme, lysine, B-lysine, which make this beta hemolytic feature of Staph aureus. And here we have our test, the sample we have, or the colony we have. We don't know whether it is Agalactiae or other Streptococci. And when we do this, one is like strict up to or closer. Urabona aho umurongo ugarukira ntabwo ugera hano kuri Staph aureus, ugarukira haruguru. Inoculation zone does not touch Staphylococcus aureus inoculation zone. But the substances or the factors that are produced by this Streptoagalactiae, which we call them as CAM factor, we allow this or we amplify this lysis of B-lysine to create a more or larger hemolysis zone. And this was only discovered in 1944 by these three people, which immediately gave the tests their name, CAM test. So the activity of Staphylococcus aureus B-lysine is amplified or is enhanced by extracellular factor produced by Streptococcus agalactiae. So here there is some factors produced by the colony here of Streptococcus agalactiae, which enhance or amplify the ability of B-lysine to create more or to expand more kill more red blood cells. And this is typically specific to Streptococcus agalactiae. Other Streptococcus, we not produce this zone. We only show like this, but we not create this this larger clear zone as interaction between Staphylococcus B lysine and in the test sample or test isolate. Is that clear? Is that clear? So if not, you can we can come back on this after your break. Let's come back on at 10:50. Come back at 10:50. Can we resume? Are you there? Hello? Can we resume? Are you there? Or can we resume? Are you there?
Yes, lecture.
I see many are no longer connected. Huh? So let's continue. Another streptococca of interest is Streptococcus pneumonia, which is normal flora in oropharynx for up to 50 of healthy individuals. So 50% is a lot. So meaning many people just have this in their oropharynx as normal flora, and it is gram-positive as say diplococci. It have lancet shaped kind of lanceolate in pairs, diplococci, or very short chains. It is non-forming, non-motile, and always capsulated. So all cells are capsulated. Catalase negative produce pneumolysine that cause alpha hemolysis on G. So it is alpha hemolytic. And its growth is inhibited by optoshin disc, which is a ethyl hydroxyprene chemical can just inhibit its growth. It can be lysed by bile. It ferments glucose to lactic acid. It has antigen and more than 87 serotype of this capsule antigen. So you can distinguish many serotypes. And tendency to lyse after initial growth, starting from the center. Doxman characters. Crater-like appearance, you see this, and the growth is enhanced by CO2, 5 to 10% CO2. So as variance factor, it have capsule, which allows antiphagostic feature. It's an IgA protease. You have this, it is able to cleave IgA. Then the bacteria can evade the immune system. protection of the upper respiratory tract by the immunoglobulin, pneumolysine, an intracellular membrane damaging toxin. So it interacts with target cell membrane to form transmembrane pores, thus destroying the cell lysis. So it degrades red blood cells. And it's toxic on human cells, a slow ciliary beating, toxic to, so this will allow colonization again as it slow down the ciliary beating. Toxic to pulmonary endothelial cells, disrupt of endothelial barrier, allowing spread of organisms from alveolar to bloodstream. It have autolysin that break down the peptide cross-linking of the cell and cell wall peptidoglycan to the cell lysis. And the lysis of cell wall will allow more fragments, which can induce huge inflammatory response, which can be the cause of symptoms that we see. As clinical condition, it's a cause of central nervous system, heart valve, bone, and joints infection, inflammation. It can spread from nasopharynx to other structure directly, contiguous structure, middle ear, sinus, lung, or hematogenously to central nervous system, causing meningitis, heart valve, or bones, osteomyelitis, and joint arthritis. It can cause pneumonia, lobo or bronchial pneumonia, and meningitis is more in the group of younger children, younger age, or older age. Can cause otitis, sinusitis, empyema and pleural effusion, lung necrosis. So it can cause exacerbation of chronic bronchitis, endocarditis, all these conditions, septicemia and bacteremia. As laboratory diagnosis, the specimen can just be from the infected site, can be CSF, cerebrospinal fluid, blood, pus, sputum. First, on specimen, gram-positive lanceolate cocci can be seen, always capsulated. You can see the capsule by using Indian ink. Culture on blood agar or chocolate blood agar. Incubate, of course, under 10% CO2. The candle jar that we have seen. So it have very small colonies, pinpoint colonies, and alpha hemolysis that are dome-shaped. Gram positive also in short chain or mostly in diplococci. I can show you the colony. So I just wanted to show you how the colony looks like. They are alpha hemolytic, and they are optogene susceptible, as we said, and the colonies have autolytic changes, starting from inside. And that is the characteristic that we said, Drogsman appearance, dome shaped. They start rising from inside, inside the cell, autolytic changes. So the outside, or it starts inside by visualization, and they are really typical colonies. And this is, sorry, I didn't include this, and I think they are important for you to visualize. We said the gram is positive, and they are lanceolate, so they are like lens, and sometimes in diplococci, and sometimes in short chains, not as the longer chain of Streptococci, Pyogenes, or Agalactiae. So they are in short chain, and or in diplococci with this criteria of lanceolate picture. Catalase negative, optogene positive, and they are bile soluble. So the bile can lyse the colony. These are two criteria to differentiate Streptoneumoniae from other streptococci. And on sputum, you can do quelling test, which is based on polysaccharide capsule antigen. And you can agglutinate this and see that you can use latex agglutination. This is more for meningitis, diagnosis of meningitis, or you can do other test serotyping and so on to diagnose. So they are susceptible to penicillin, to most antibiotics. Some few are beta-lactam resistant strain, which are increasing now. You can use ceftriaxone, cefotaxime to treat the patient. And based on minimal inhibitory concentration, you can on penicillin. So in the lab, the susceptibility testing allows you to know at what amount or the concentration of drug that inhibits the growth of bacteria. And that minimal concentration that inhibits growth of bacteria can allow you to know whether the antibiotic can treat or not. So when the minimal inhibitory concentration of penicillin is less than 0.1, you can treat the patient easily with penicillin and ampicillin. When it is in a range of 0.1 to 1, the penicillin will not work. So we have to use ceftriaxone, cefotaxime, especially for meningitis, and sometimes high dose or ampicillin for other conditions. So by increasing the dose, you can be able to treat, but for the meningitis, best to go for this. And when the MIC is higher, up to 2, MIC for penicillin, so you will have to combine vancomycin and rifampicin. Sometimes a high dose ampicillin, and so especially fluoroquinolone in non-meningocytes, in other conditions except. And this MIC is important to know which drug or which combination of drug you are going to use to treat patients. So there is some, yes, please.
Excuse me.
Yes.
What is the meaning of MIC?
Who can answer the question? MIC. Who can answer the question? Okay, James. Is it James? MIC means minimal inhibitory concentration. So that is the lowest concentration of drug that inhibits the growth of bacteria on media. So when you have your isolate, your growth, you can be able to see what is what is the minimal inhibitory concentration of this isolate. So by increasing gradually the concentration, you can be able to know this isolate or this strain can be inhibited only by 0.1 milligram per liter of the antibiotics, or in between here, or even upper concentration. So the minimal inhibitory concentration is linked to the isolate, to the strain that you have isolated. So when the growth can be inhibited, completely inhibited by less concentration, that is lower than 0.1 milligram per liter of penicillin, then penicillin and ampicillin can be used. In between, or between 0.1 and 1, then you don't use penicillin because this will not be useful, especially in meningitis. You have to use cefotaxime and high dose penicillin or ampicillin for other conditions. So you have to increase the dose of penicillin. And with MIC higher than 2, that is minimal inhibitor concentration of 2 or higher milligram per liter of penicillin, then the penicillin will not be useful completely. So you have to use other drugs, including vancomycin, rifampicin. Is that clear? Wabajije ikibazo yakirize.
Yego.
So as prevention, there is a vaccine. We have a vaccine that have 7 variants. A question? Seven variant pneumococcal conjugate vaccine, which combine different most available serotypes. We have some other pneumococcal conjugate vaccine that combine additional serotype, up to 23 valent and conjugated polysaccharide vaccine. So you can use this kind of vaccine to prevent the meningitis, especially in case in case of epidemic driven by this streptococcal pneumonia, meningitis driven by this. Any question? Virulent streptococci, known as oral streptococci, also. There are 5 distinct groups. In most cases, they are normal flora of the although they can cause some problem, dental caries and so on, but they are just normal flora of the oral cavities. And we have these 5 groups. They are part of normal flora of the mouth, upper respiratory tract. So they inhibit colonization of many pathogens, including pyogenic streptococci, by producing 2 components. They produce bactericin and oxygenated oat to inhibit colonization of many other pathogens, including this one. So the protecting oral cavity from pathogen. However, sometimes they can cause problem, like they produce polysaccharides, which is found in dental plaque, which leads sometimes to dental caries. They can enrich blood after dental surgery, causing infective endocarditis. They are able to produce acid like this one, which is important in caries, dental caries. They can produce fibronectin and can allow them to adhere to the heart valve, causing colonization and problem. So as the most important clinical condition, there is dental caries, which is caused by mutant group, gingivitis, pyogenic oral infection, subacute bacterial endocarditis, with patients with abnormal valves, and can reach bloodstream after dental surgery only. Otherwise, it is not able to cross and reach there. Imitis group, and they can sometimes cause brain abscess after dental surgery again. Lab diagnosis, specimen from the site, infection site, blood, pus, or aspirate, gram stain, gram-positive cocci in chain, culture, they are majority alpha-hemolytic or some non-hemolytic. They are all catalase negative. They are optochin resistant and bile insoluble. And this criteria allows them to be differentiated from the previous genera, the previous species, and cannot grow in this. We see that this differentiates them from cocci. And you can use other tests, API streptococci, to identify which genera or which species is implicated. Do you see my slide? Do you see the slide?
Yes.
Yes.
So in most cases, you have to give prophylactic penicillin to the patient with dental surgery, especially to limit the risk of all that dissemination that you have seen. Can use penicillin and other beta-lactam. Sometimes you have to combine. There is synergy, and the vancomycin can be used in case of allergy to penicillin agents. Any question with this? They are mostly normal flora of the oral cavity, and they can cause problem, particularly when there is an intervention like dental surgery, or sometimes they produce this huge amount of acid and cause caries, dental caries. Any question on virulent streptococci? No. We have Enterococcus, which is another genera, different from streptococci, staphylococci. And in here, we have Enterococci fecalis, Physium durans, but fecalis is the most common, or what we see as the cause of some disease. It is normal flora of the gut and can be found in environment. They are cocci gram-positive and mostly occur in singly or pairs or in small chains. Formerly classified as Streptococci fecalis, but now they have seen that it's a different genera. It can grow in hypertonic saline or bile, example, Macon medium. So this we then look like Staphylococcus because Streptococci are not halophilic. And having this halophilic condition, we allow this to grow in this hypersaline media. They are also resistant to bile. They cause urinary tract infection, and that is the most common. They can induce bacteremia, subacute bacterial endocarditis, intra-abdominal pelvic infection, including peripheral sepsis, wound infection, and others, meningitis, neonatal sepsis. As diagnosis, the specimen will depend on the affected site, and direct gram stain, gram-positive cocci in chain and short chain, culture on BA, Macon medium, incubate 24 hours, 18 to 24 hours. And you can see variable hemolysis pattern. They can be beta, gamma, or alpha. So they don't have a fixed, they can grow on Maconke and lactose fermenting. They are gram positive, as you say, in pairs or short chains. How do we differentiate them from streptococci? They hydrolyze esquilin. This is very important. So of course, they are catalase negative, but they hydrolyze esquilin, and this differentiate them from either staph or other streptococci, and they hydrolyze also pyridonyl at arylamidase. So they produce this enzyme to hydrolyze this chemical, and they can agglutinate this streptococcal group antigen. So as susceptibility, penicillin, they are always resistant to penicillin, they are always resistant to enterococci, and late we have seen that linezolid resistant strains have appeared as well, so causing trouble. They have intrinsic resistance to aminoglycosides. They are susceptible to ampicillin, gentamicin, and linezolid sometimes. So it is imperative to do susceptibility testing. So by combining several agents like penicillin and the aminoglycosides, streptomycin, you can be able to treat some of these, but it's important to do the susceptibility testing to confirm your susceptibility pattern before treating the patient. Streptococca, these are strict anaerobe, variable hemolysis. They can have beta, alpha. They are normal flora of mouth, upper respiratory, but vagina, skin, and they are strict anaerobes. And they are part of mixed anaerobic, they can take part of mixed anaerobic infection, and mostly multiple bacteria. And they are opportunistic pathogen, causing brain, lung, abdominal, and pelvic abscess. But in multiple bacteria problem, like complex, not as individual bacteria. And these are the only strict anaerobe cocci, streptococci, that can be described in here. And treatment can be a combination of metronidazole, penicillin, clindamycin. And as this is in group of bacteria, so normally the combination of antibiotics is preferred to clean all pattern of resistance. Any question so far with all cocci gram positive? So we have seen Staphylococcus, we have seen Streptococcus, We have seen Enterococcus. And finally, we have also seen the Streptococci. These are the most important gram-positive cocci, cause of disease in human. Izo tuvuze ni izo majority zikora ziteza indwara ziri gram-positive cocci. Is there any question? Any question is before we start Neisseria? If no question, let's move forward. Neisseria is a family of gram-negative cocci because of human disease. And we have 4 genera. We have Neisseria, Moraxella, Acinetobacter, and Vernella. So those are the 4 genera in the family of Neisseria that we are going to see now. Sorry. Let's start with Neisseria. Neisseria are commensal of mucosa, so they are part of the commensals on mucosa, and approximately there is 11 species that colonize human mucous membrane, but only 2 are pathogenic. So only 2 species are pathogen, and those are Neisseria gonorrhea, the cause of the gonorrhea, also called gonococcus, and Neisseria meningitidis, the cause of meningitis, also called meningococcus. So those are the only 2 pathogenic species in the 11 species of Neisseria. They are gram-negative diplococci with their agent site flattened, like cough beans. So the agent site of maybe by showing the image, we make it much more easier. Not very clear to me. Maybe you have to. So the one side is flattened and they are adjacent to each other. So if you can see in gross, you can see that one side is flattened and they are like coffee or beans of coffee, kind of, or and they have this gram negative. So diplococci adjacent like kidney-shaped or coffee beans, they are aerobic. They have strong oxidase positive. They have catalase enzyme. They are susceptible to drying, very susceptible to drying condition. So meaning the sample should be quickly be inoculated on media, otherwise you lose them. They are non-motile, non-spore forming, and require 5 to 10% CO2 for growth. They are fastidious. They require some enriched media like thiamat media, like chocolate blood agar media. Their growth is inhibited by toxic metal and fatty acid. Thus, they are really fastidious. They are encapsulated. Meningococci are encapsulated. They have capsule, and meningococci ferments glucose and maltose, while gonococci ferments glucose only. They don't ferment the maltose. So they can be found inside the cell in neutrophil, polymorphonuclear, in especially inflammatory exudates. They are intracellular most of the time. They contain endotoxin in their outer membrane, like the meningitis or meningococcus have a lipopolysaccharide. We have seen that's gram negative. And the gonorrhea or gonococca have lipo-oligosaccharide. 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 Both organisms may bear fimbriae, so these are adhesion organ. You know them, and both pathogens produce IgA protease, which promote virulence. So these are kind of they are kind of virulence factor, and sorry, some interruption. sorry. So we are saying that both organisms, either meningitis or gonorrhea, have fimbriae, which allow them to adhere on the surface, and they both have this IgA protease. You know, IgA, or you will know this antibody is a most or the protective antibody of the mucous membrane. Huh? So normally this is located on the mucous membrane and protects invasion of bacteria, viruses from the mucous membrane. So it has this protease that cleave or destroy the IgA. So thus allow this bacteria to colonize the mucous membrane. Mm? And this promotes the virulence of this bacteria. So I think we have seen a better image, but as we said, there are sometimes intracellular, they are inside of polymorphonuclei. So let's start with meningitis, Neisseria meningitis, which is primary asymptomatic colonizer of the human nasopharynx, and up to 10 people, or 10% of population has already this in their nasopharynx. So you can see many people has the pathogen. And it's causative agent of meningococcal disease, which kills an estimated of 50,000 people worldwide annually. Not too many, but we see that it can be factor cause this in some specific region more than others. In this case, Neisseria meningitis disseminate from the nasopharynx into the bloodstream, where they can survive and divide, giving a bacterium which can involve meningitis, which is the most, and sepsemia. And this is the two important conditions associated with Neisseria meningitis, meningitis and sepsemia. So it is grouped on the basis of meningococcal capsula. So we have it has many subtype cell groups up to 13, and A to all these, and 6 of which can cause epidemic. A, B, C, W1, SAT5, X, and Y. They are the most cause of epidemic, the outbreak. Group A and C, and less commonly B, being responsible for epidemic. Most the meningococcal population is classified in different phylogenetic group. The clonal complex defined by the locus sequence typing. So this is a more genetic classification. As virulence factors, they have protective polysaccharide, capsule, which is very important to survive in the bloodstream. They have the endotoxin that we have seen, lipopolysaccharide, LPS. They have pillars that allow them to fimbriae that allow the adhesion. They have this IgA protease that cleave the mucous membrane antibody, thus allow colonization. And they have protein capable of binding complement factor H. So by this, then they can inhibit the activation of complement. They can acquire iron. So as clinical findings, this is meningococcemia, septicemia of Neisseria meningitis. You can see how much it's really also called Waterhouse-Friderichs syndrome. There is high fever, shock, disseminated intravascular coagulation, thrombocytopenia, adrenal insufficiency, and followed by death. So when the pathogen disseminates, then the death will just come quickly. and they are also cause of meningitis. They arrive in brain and cause with meningitis. You have fever, headache, stiff neck, increased pulmononuclear in spinal fluid. As epidemiology, we have seen that up to 10% of population just have this as natural in nasopharynx inhabitant of L. meningitis, and they spread through or via respiratory droplets. Carrier are asymptomatic, and in older children and young adults, they are most carrier. These high people, the carrier are high people who live in close quarters, like we have military recruit, college of students in dormitory, and meningococcal meningitis occur both sporadically, so they happen in a sporadic way, many group B and C, and in epidemic way, especially for group A, with highest incidence during the winter or early spring, that is in Europe or countries. So as diagnosis, maybe, sorry. So there is what is called meningitis belt. The meningitis belt, where most patients comes from, and this is stretch from Senegal to Ethiopia. So this belt, this is where up to 65% of all cases in Africa are just reported in these few countries: Burkina Faso, Chad, Ethiopia, Niger, and Senegal here. So this stretch or this belt, so that is where the most epidemics takes place. They are seasonal. As we said, during the dry season, between dust winds, cold night, and upper respiratory tract infection combine to damage the nasopharyngeal mucosa, increasing the risk of meningococcal dissemination disease. So dry season combined with cold night with winds, sometimes upper respiratory tract infection, we allow this normal or the commensal of our nasopharynx to disseminate, to cause the meningococcal disease. As diagnosis, the gold standard is culture. So there is no, and you can isolate Neisseria meningitis on chocolate agar plate. Further testing to differentiate species include testing for oxidase, catalase, and fermentation of glucose and maltose to differentiate species. You can do serology by typing agglutination or PCR to identify quickly the pathogen. As treatment and prevention, you can use penicillin. They are sensitive to penicillin, chlorphenicol, or a third generation cephalosporin like cefotaxime or ceftriaxone in person with problem with allergic problem with penicillin. You can also use rifampicin for children and carriers to eliminate the pathogen. Meningococcal polysaccharide vaccine are available, so either in bivalent, so that is preventing for two groups only, trivalent or tetravalent. So with this, you have like protection from A, C, Y, W, SAT5. So, and this is mostly deployed when an epidemic of Neisseria meningitidis takes place. So, when there is identification of this at the lab, then the outbreak group can immediately try to vaccinate people in the area to make sure you are protecting the population. Any question with Neisseria meningitidis? Any question? Any question? If no question, let's also discuss Neisseria gonorrhea, the cause of gonorrhea, the causative agent of gonorrhea, which remain the most common bacterial sexually transmitted infection, as more than 60 million new cases are reported globally. So you can imagine 60 million cases at ERA basis. This is too many. So that makes gonorrhea to be the most common bacterial sexually transmitted infection globally. And it takes all over the world there, either in developed country as well as in. So as variance factor, we already seen that pili play roles by allowing adhesion, attachment, but also antiphagocytic role. They have lipo-oligosaccharide. We have seen that endotoxin in the outer membrane. They have IgA protease, which hydrolyze secretory IgA. They have purine A, which the purine protein in the cell wall inactivate the complement, which is important to destroy the pathogen. So as clinical manifestation, we unfortunately in women, the patient is or disease is in most cases asymptomatic, which allows more dissemination. So in some cases there is a purulent vaginal discharge, but in most patients there is no symptom. The asymptomatic carriage is common. Infection is primary in the cervix, causing purulent vaginal discharge, lower abdominal pain, and abnormal bleeding. Vaginal bleeding may also infect the urethra, vulva, and rectum. It can ascend to fallopian tube, causing the acute inflammation of the fallopian tube, salpingitis, ovarian abscess, pelvic inflammatory disease, and high probability of sterility. As people will not notice that they have this problem, the women will not notice as it is more asymptomatic, but it will cause all these conditions of. So sometimes it can cause ectopic pregnancy, which can be a result of scarring of the tube, damage of the fallopian tubes, allowing the eggs to drop and sperms also to find, and the fecundation can just take place outside and then so for men, asymptomatic infection is rarely seen. So in men, the symptoms will always be there. So you can see purulent penile discharge, especially at start, acute urethritis, dysuria, purulent penile discharge, increase in urination frequency, and sometimes the main will be the area where they will know that the infection is there. So by diagnosing the man, then you can be able to trace the origin of infection and as well treat the couple, treat the partner, because this is not seen in most female. If untreated, can result in epididymitis, prostatitis, and erythrostructure infections. And people practicing oral anal sex we result in oral anorectal gonococcal infection. It can cause all those problems in oral cavities, in rectum, leading to period discharge, burning pain, sometimes blood in stool. For babies, there is two important conditions: ophthalmia neonatal, the purulent eyes, discharge, tearing, and orbital edema, and this is mostly caused by during the birth. The children, a few days after birth, the infection just comes from the mother and can be as damaging as this. And conjunctivitis can be in even adults, which is result of poor handling of infected genital secretion. If you don't manage well the discharge, cleaning, or washing hands, it can result in infecting the eyes, leading to conjunctivitis. And if untreated, they can evade bloodstream and cause disseminated gonococcal infection, which is characterized by hemorrhagic skin lesion, fever, and chills. So, and this is more dangerous or life-threatening condition with dissemination. One of the condition, disseminated gonorrhea condition, is arthritis. You can see how the patient can be affected in all area. As diagnosis, with colony oxidase positive, gram negative diplococcus, catalase positive, you can already think about Neisseria gonorrhea, but confirmatory tests are more biochemical testing, serological testing. Sometimes you can use fluorescence antibody test, specific fluorescence antibody test, colorimetric test, or even PCR. Treatment, not such very complex. Uncomplicated infection can only be treated by cephalosporin or fluocinolone plus doxycycline, erythromycin, amoxicillin. So, in case of gonococcal conjunctivitis in newborn, which is more prevented mostly often by use of erythromycin ointment. So, especially when the mother is infected, you can already prevent this in newborn. So, by using this ointment of erythromycin, then you can prevent the conjunctivitis in newborn. Any question with Neisseria gonorrhoeae? Any question? So if no question, next genera is Moraxella, which is also cocci, diplococci, also cocobacilli. So they have this cocci in between cocci bacilli morphology. They are strict anaerobes and oxidase catalase positive. And the major pathogen is called Moraxella cataralis, previously named Branamella cataralis. It is found in nasopharynx as commensal and is transmitted by respiratory aerosols, as does meningococcus. It has lipo-oligosaccharide, which is considered as variance factor, and cause disease in immunocompromised patients or patients with any other underlying chronic condition, like chronic obstructive pulmonary disease. So they are then opportunistic infection. They are opportunistic pathogen. Only those immunocompromised or patients with underlying chronic disease, we only have problem with this. And they can cause otitis media, sinusitis in children, laryngitis, bronchitis, bronchopneumonia in adults, but all we specifically comes in immunocompromised patients or with some underlying conditions. As lab diagnosis, specimen will depend on location. Gram stain, we have gram negative cocobacilli, which may be intracellular. Culture on BA or chocolate blood agar, you can use 5% CO2. And as biochemical reaction, they are oxidase positive, do not ferment any sugar, carbohydrate, and this will quickly differentiate them from Neisseria or Neisseria gonorrhoeae or Neisseria meningitidis, especially as meningitidis ferment both maltose and glucose. So, in question with this, I think it is not such a very typical pathogen, but you just have to know this. Acinetobacter is also cocobacillary picture rods. They are strict aerobes, oxidase negative, catalase positive. They are also opportunistic pathogen associated with mostly nosocomial infection and cause disease associated with respiratory therapy equipment and dwelling like catheter, and they form biofilm on the equipment or the internal or external body. They can cause periodontitis, bacteremia, urinary tract infection, sepsis, pneumonia, but all these are related to the immune system of the host. They can be found as normal flora of the skin, respiratory, and intestinal tract, causing sometimes genitourinary tract infection, UTI, as we said. Acinetobacter we grow on routine laboratory, including sheep blood agar colony, and they are mostly isolated. They are resistant to many classes of antibiotics, and treatment will be based on combination of antibiotics And rifampicin is mostly used in treatment of post-neurosurgical Acinetobacter meningitis. So you use rifampicin. And carbapenems can also be used in treatment of Acinetobacter. So you will see when you do your lab tour that Acinetobacter is not rare. It is really commonly isolated. And and should be well identified, as well as susceptibility testing done, given its feature of resistant to many antibiotics. Another genera that we see as gram-negative cocci is Kingella, and the most pathogen is Kingella kingai, which is normal flora of the throat, especially in children, transmitted via respiratory secretion. It has pili and exotoxin, and cause infection in young children. Septic arthritis, osteomyelitis, bacteremia, endocarditis. Occasionally can cause lower respiratory tract infection, including meningitis. But in adults, it should be immunocompromised individuals. So it can cause urinary tract infection, respiratory infection, septic arthritis, and we treat with beta-lactams, tetracycline, erythromycin. So it's such also brief and mostly normal flora of the throat, especially for children. And lastly, for the gram-negative cocci or Coccobacilli, we have Veronella, which is completely anaerobic gram-negative diplococci, and also part of the normal flora of mouth and the colon, vagina. It's rarely opportunistic pathogen and can cause abscess of sinuses, tonsils, and brain, but always opportunistic. Also, minor component in mixed anaerobic infection. And only Veronella parvula and alcalensis are associated with human disease. So the two species are associated with human disease. So these are a category of gram-negative cocci which can be involved in disease in human. So we have Neisseria, we have Moraxella, we have Kingella, and we have Veronella. So as gram-negative, those are the most important gram-negative bacteria, cocci gram-negative, that can cause problem in human as disease. Any question? Any question? Can you hear me? Hello. Any question? If no question, we can limit or stop our lecture here. And as said initially, we have other bacteria. We continue with bacteria. Now we have this group, cocci completed. So we have the rods, gram-positive rods, and gram-negative rods. And I will provide you or give you the lines for presenting in this area for group presentation assignment. And we continue to other pathogen while complementing from what you presented in your assignment. Class representative, ngira ngo dufite 5 group in GACO and 12 group in Huye. Am I correct? Is that correct?
You are right.
So I will provide topic for the 5 group, each group with a topic, and for the 12 group in Huye, each group with topic, and you will have to present. You will have to prepare the slides and present. We have like a session of 10 to 15 minutes each group and 5-minute questions. So 20 minutes in total for each group. And we have that during our next lecturers for bacteriology. But before we move with the assignment, on Monday, we will also progress in virology. In such way, we also reach the area of possible assignment topic for each group. Is that okay? Hari ikibazo? If no further question, niba nta kindi kibazo twagarukira aho ngaho. I will try to send you the notes, either virology and this that we have completed. Sorry, I had a lot of disturbance, a lot of things ongoing, but I will try by now to send you the assignment. Sorry, the notes, and I will prepare the assignment that I will send to your class representative, and I will give you some hours to work on those assignments. Sibyo? Nzabaha some of the days kugira ngo mukore kuri izo assignments, while I will also having some other responsibility here. Is that fine with you? Nta kibazo?
It's fine, sir.
Okay, so nice day. We
Yes,
continue
teacher.
on Monday, same time, and I hope by then I will already prepare your assignment.
Yes.
Thank you.
Yes.